So thank you very much for giving me the opportunity to talk about asthma and sphingolipids. The first slide shows that this is a group effort by many, many participants at Weill Cornell, at Columbia, at the Brigham's, downstate, and funding luckily from several institutions. So we are concerned about asthma. Asthma is a leading serious chronic illness in children. 300 million are affected worldwide, and patients can die from asthma. Treatment is symptomatic because the pathogenesis is not really known. There are heterogeneous phenotypes, and most importantly, there is a genetic predisposition. As you can imagine, with such a significant disease, lots of research is going on. Independent of the different phenotypes, endotypes, genotypes, there are three relevant findings that are found in all asthma. It is inflammation, it is smooth muscle hypertrophy, and here on the right side you see a very extreme case, and you have airway hyperreactivity. So Asthma GWAS have identified the 17q1221, and it's actually the most highly replicated and significant asthma locus. The first Asthma GWAS dates back from 2007, and they identified 17q, and the original paper had 3 genes on there. By now it has been extended to 10 genes. And the original paper showed that genetic variants on 17q1221 that increase ORMDL3 expression contribute significantly to non-allergy childhood asthma. That's 2007. And in 2010, the Weissman group and the Chung group showed that ORMDLs affect sphingolipid homeostasis and protein quality control. So these are very important papers that all of a sudden link ORMDL to sphingolipids, and ORMDL has been linked by the first GWAS to childhood asthma. And this is basically a simplified model that has served us to structure our experiments. So you have ORMDL, it increases sphingolipids, decreased sphingolipids will eventually turn down ORMDL, turn on sphingolipids, etc. Feedback mechanism. And our question that made us enter this field was, what's the effect of decreased SPT activity on airway reactivity? And so we could show that if you have decreased SPT activity, it will lead to increased contraction. And we showed this in many models and in many different ways. And I'm summarizing here more or less. The large airways, we used bronchial rings for this. They were from SPT-LC2 haploinsufficient mice. So these are not knockout mice, but these are mice that have just less SPT activity and thus less sphingolipids. We also treated mice with myriocin, and we took bronchial rings and based them in myriocin. And common to all is that whenever you have less sphingolipid synthesis, you have increased contraction. And this is not only true for large airways, it is also true for small airways, which you see down here. If you take precision cut lung slices from these SPT-LC2 haploinsufficient mice, compared to controls on top, the SPT haploinsufficient mice on the bottom, and you expose them to methacholine, but that's the classical drug to induce airway contraction, you see that the SPT-LC2 mice hypercontract. And given all these results, our model was updated as such. We know that decreased sphingolipid synthesis causes airway hyperreactivity. And we know that if we look and we focus entirely on decreased sphingolipid synthesis, we don't have any indications for increased inflammation, airway remodeling, or mucus production. So decreased sphingolipid synthesis is not responsible for these findings that go along with asthma, but it's responsible in our model and our hypothesis for airway hyperreactivity. And based on this observation that low sphingolipids are found in cases with airway hyperreactivity, our hypothesis was that serum sphingolipids are decreased in asthma. And in order to investigate this hypothesis, we worked with the Columbia epidemiology group, Matt Krzanovsky, who leads a large New York City neighborhood asthma and allergy study. And we had 243 young kids aged 7 to 8, of which 152 had asthma, and the same patients, 147 of these asthma patients and the controls were also followed up at age 10 and 11. And our results showed that sphingolipids are not decreased in asthma. On the contrary, we found in plasma increased dehydroceramide C18 and ceramide 20. And we could show that if these two parameters were increased at age 7 and 8, they predicted the persistence of asthma at age 10 and 11. So very interesting result, but absolutely not fitted to our original hypothesis or to our model. We then moved on and investigated the question in a new study whether sphingolipid de novo synthesis is altered in pediatric asthma. And for this, we designed a larger study that eventually included 120 patients, and of which 61 had asthma. Kids were from age 5 to 17, and they were recruited in Manhattan, this is here from Google Maps, and in Queens, because these are completely different populations, and thus we would have more insight and a better study plan. We collected whole blood, plasma, PBMC. The entire asthma diagnosis was physician guided. We accounted for the presence of allergy by measuring IgE and eosinophils, and we measured 5 relevant SNPs. They all orient in this cluster here about GSD, and OMDL, of which the 72, we did 2 things. We measured sphingolipids in plasma and whole blood, and measured de novo synthesis by incubating PBMC from the kids. We incubated the PBMC with stable isotope serine and measured then incorporation into sphinganine and sphinganine 1-phosphate. And before I go into the results, I need to walk you through the result format, because otherwise it's an overwhelming amount of data and doesn't seem good. So for each sphingolipid, we have a little box, and we stratified by control, blue, yellow. Furthermore, we have so those is characteristic of a non-allergic type, and then we have controlled and asthma patients with high eosinophils. So each box shows you on the left side the non-allergic asthma type in yellow compared to a non-allergic control, and on the right side you have an allergic group, of which one group has asthma and the others have eosinophils for something else. The means are compared by a 2-sample t-test. But with such an amount of data, you can't go by means. You need to correct for false discovery rates, and so we used the Benjamini-Hochberg procedure, and that generated a critical value, and everything below that critical value can only be called significant, and the significance is marked here with an asterisk. The next slide shows you the sphingolipids in plasma. So the big picture here is a summary below. In plasma, we saw increased dihydroceramide C18, increased dihydroceramide C21, increased ceramide C20, and increased sphinganine, most eosinophilic groups. So an asthma patient with high eosinophils, that means an allergic asthma patient, has dihydroceramide C18, C24, sphinganine, we had previously seen in the epidemiological study. But we now apply strict and appropriate statistics, group, these differences did not become statistically significant. Of note, groups, so here we have 120 patients, in the other groups we have 240. It has significant results when we looked at whole blood. And here is the same and for ease of viewing, I put a red dot for all the significant differences. So in the dihydroceramides, 18, 18, 22, 24, 24, 1, and the sphingomyelins 18, 18, 1, 24 are always significantly lower in patients with. So in the end, the finding is here, control patients with non-allergic asthma have lower sphingolipids. Once the patient has allergic asthma, sphingolipids are significantly higher. And that's basically the whole message. This difference doesn't exist in the control group, whether you have low or high eosinophils, there is no difference in your sphingolipids. So this is a very important finding because it's the correlate of inhibited sphingolipid synthesis and lower sphingolipids finally in some kind of cell type. We also looked at the SNPs and their effect on whole blood sphingolipids. And the classical SNPs 72 and 807 both correlated with decreased sphingolipids, and that was a risk allele dependent. So more risks alleles, lower sphingolipids. There was no correlation in this group, the other SNPs, however, it correlated, but it didn't reach significance. So I would propose to say, let's not exclude these other SNPs. I think the study group ought to be bigger to make that final assessment. We then looked at sphingolipid de novo synthesis by looking at the whole group and then correlating with SNPs. And the finding is that all patients with asthma, independent of allergic or not allergic, had decreased de novo sphingolipid synthesis. And looking at the SNPs, we found that also probably due to group size, only the risk allele for 72 correlated significantly with decreased sphinganine 1-phosphate synthesis. You see here on the right side. So very important finding that there is an inhibition of de novo synthesis measured by stable isotope incorporation. All the data I've shown you so far are far removed from the lung because PBMC, red blood cells, who knows, maybe they have nothing to do with what is going on in the lung. So we then obtained primary airway epithelial cells from adults because it's really not feasible to obtain this from children. And we looked at different types and stratified them by the risk allele for 72. And indeed, we found in the risk allele T for 72, decreased sphinganine and decreased dihydroceramide, when at the same time we didn't find any differences in ceramides that originate from, that can also originate from the recycling pathway. So that's the end of my presentation. And in summary, I can say whole blood sphingolipids are decreased in non-allergic asthma. This is a first finding. And in the entire group, sphingolipid parameters in whole blood correlate with risk alleles in SNPs 72 and 807. We have decreased sphingolipid de novo synthesis measured in PBMC in pediatric asthma patients, and again, a correlation with risk allele TT for SNP 72. And we find decreased sphingolipid de novo synthesis in primary airway epithelial cells carrying risk alleles. And so our model persists. We want eventually to translate this into something beneficial for patients. And we can certainly, our data are consistent with the finding that if sphingolipids are low and sphingolipid synthesis is low, the likelihood of airway hyperreactivity is high. We don't have a model in our approach for inflammation, airway remodeling, or mucus production. But next steps are obviously to understand if we can, by some means, prevent hyperreactivity. So thank you very much, and be happy to entertain questions.